next, we have Mike Stadnisky from Phinotex, upgrading your panels on any flow cytometer with NovaFluors.
Great, thank you. Let's make sure this actually works there. Figured we'd all been surviving through lots of PowerPoint and Zoom. So I wanted to give you a brief brain break with a story. So this beautiful evening, this Emperor Akbar, who was a Mughal emperor in the 16th century, strolling around his beautiful gardens with his entourage. And he was in a good mood and he was in a good humor, and he always liked to test his courtiers. And so he said, I have a challenge for you. So he took out a sword and he drew a line in the ground. He said, do you see this line? And his courtiers, who were mostly sycophants, said, yes, and what a beautiful line it is. And they said, okay, be that as it may, is there anyone here who can make this line smaller? Anyone who can do this will receive a reward from me. Sort of used to the emperor's test, right? The courtiers all scratched their head and they couldn't find a way to do it. And so he brought his minister Birbal to the garden and he gave him the same challenge. And Birbal unsheathed his sword, and with the tip, he drew a line that was twice as long as the one that was next to the emperor's line. And he said, there, Your Majesty, this is what you've ordered me to do, and I've done it. And he bowed courteously. He said, the line you drew is a much smaller line. And I share that story because, one, it's good brain break, but two, because we're going to talk quite a bit about this today in terms of how we can leverage new dyes and leverage the capabilities of our instruments. And I also wanted to kind of bring this back to the first commandment of storytelling, right? Since we have to give these short talks, the greatest commandment is make me care, right? Emotionally, aesthetically, especially for us scientifically, make me care. We know what it's like to not care, right? We're bombarded with information, and I think it's it's tough, right? We have to just sort of say, well, what's going to make me pay attention? The first way to do this, for what it's worth, going meta for a little bit, is we're going to start with the ending. So I'm going to start with the ending of the presentation, which I'll bring back again at the ending. So I'll start with the ending. We were able to plug and play 6 labels into a panel that's actually already been discussed today, into this 35 color panel, of which 34 of the colors are commercially available. We're able to phenotype 32 different populations, look deeper into the biology there by leveraging some of our NovaFluor reagents on chemokine receptors and other phenotyping marker. And we've put this all together into conjugation kits, which are actually competitive with antibodies. And so you can actually purchase a conjugation kit for the same price as you would get, an antibody, right? And it's not done on tests; it's done on actual mass. And so we'll come back to that topic in a second. So the second part of this is, and yes, I have my dog with me, is leverage this potential energy, right? So how do we leverage that potential energy, right? If you think about it, a story and what I've just shown you, the ending is like a pebble in a slingshot. And so let's let that go and see what we can do. And the question I always like to ask is, what if you could design a fluorescent label and you could start from scratch, right? You wouldn't start from something that's already messy, maybe say PE or APC. What if you could start all over again and work on that as an infrastructural problem? And given that, what would panel design be? And so really, we'd pull out 3 different things. We'd say we really want our labels to be both bright and clean, and we want them to really be silent on the violet, right? Because if we have, a signature, we're contributing spread, regardless of our ability to distinguish them. We really want to have them hit detectors cleanly. And we want to do that across all flow cytometers, right? So how do we think about that? And we'll talk about that in the context of all flow cytometers in a moment. We'll also mention about limiting cross-laser excitation. This is something that really should be foundational when we think about designing new dyes. The second part of this is about the practical use. How do you make a label that's incredibly consistent and also very, very stable, right? And we'll mention and show some data on that as well. And the third part of this is controllable brightness. How do you actually leverage a new-to-world capability around our ability to tune up and down the brightness of a label? And then how would you think about that in the context of panel design? The foundational technology that we rely on to do this, we actually take small organic dyes and bind them onto these single-stranded oligonucleotides. And we can then assemble them into these phyton structures, these cruciform structures. That provides us an engineering platform where we can design new fluorescent labels. And the reason we're able to do that is, we can take dyes and actually assemble them geometrically to give incredibly efficient transfer, right? That's what gives us the ability to do tunable excitation and emission. We also have 4 arms off of this phyton, which we call clusters, and that's what gives us the ability to tune up and down the brightness. This also gives us a really flexible platform for targeting. And so what we're able to do is put a single-stranded oligo off of the end of this platform and then use that to conjugate it on antibodies. Again, something we'll come back to towards the end about how we actually perform that conjugation. And we'll also mention one other thing, right, which is that we're relying upon oligo manufacturing to do this. And so it becomes an incredibly consistent platform. So let's first touch on this notion of bright and clean. I think the thing that we would all pull up immediately say, this is what's really important, that if we were to start over again, you know, the most ideal dye would only hit that detector, one detector at a time, contribute no spread anywhere else, right? That would be the ideal dye. And so, you know, we've been striving towards that. And it's the way we think around fluorescent label design. And in the first 6 labels, we've thought exactly about that in the blue, yellow, green space, a place that's seen really no innovation for decades, right? This is where our PE and PE tandems live, as we'll discuss. And what we've thought about is actually separating the excitation of both the blue and then the yellow, right? So we can actually use these together in the same experiment. And I'll show you some panel designs that actually enable that on really any flow cytometer in a moment. So you can see we can uniquely essentially excite off of the blue laser and then use these lasers independently in these detectors. Again, I'll show you an example of this. A good example of the kind of thing we're thinking about, particularly in the context of PE tandems, is what happens with something like PE dazzle, right, which is leading this decidedly more colorful life, right, and which is cross-excited off of the 488 to 532 and the 561 line. And what we've done is say, let's separate that. So I've overlaid here our Nova Blue dye, right, which we can excite off of the 488 line, and the Nova Yellow 610, which we can separate off the 561. And for what it's worth, you know, this is also the paradigm that we use for making all of our new fluorescent labels, of which we have a large number that are actually coming out at the end of June, something that I'll mention in a moment as well. But we like to think about it on a full spectrum basis about how can you do this and limit cross-excitation and limit spread across the entire spectrum. These spectra, for what it's worth, are also up so that you can examine these in the context of your panel design. We won't talk a lot about panel design because I want to really touch on some of the infrastructural elements of why this is, how these can be incorporated. We've published them up on an open resource called FPBase. They've also just this week been added to Fluorofinder, and the folks at Cytometer were really kind to put these up also on their full spectrum viewer. And I think it gives you a sense of what we're up to about where we're hitting these detectors very, very cleanly, right? Particularly off the blue and the yellow green, particularly if you look at this on their really nice spectrum viewer. I think the visualization, though, that to me is probably the simplest, but maybe perhaps the most meaningful, is what we did is we actually took single stain controls with these 6 fluorophores. They're all on CD4, compensated them, right? So you'd expect to see some spread, and then just concatenated them to see, okay, well, where are we adding spread just as a visualization, right? And how does that look like relative to this double negative population? Because when you get spread, you get the increase in this positive population. And you can see, I'm not gonna claim we have 0 spread, but very, very minimal spread, even off of adjacent channels, right? And I think to me, that's what's really important. And we'll talk about this notion of, well, what does that mean in the context of doing some biology, right? And can you separate populations well? But to me, a very powerful visualization that shows, the impact on having something that's very, very clean, right? On the ability to not spread and make the data analysis part of this easier. To that end, though, how do these compare to fluorescent labels that are out there now? So what I'm showing you is some performance testing that we've done on a FACSymphony that's at the NIH. And what we're looking at here is just a detector. This isn't a ribbon plot. We're looking at 5 detectors off the blue, 8 off the violet, 7 off the UV, 3 off the red, and 5 off the green. And so we're able to do, and we're looking now at sort of apples to apples comparisons with other labels that have the same primary detector as our label, so that this is just 2 of the labels. So we compared Nova Blue 610 to this Brilliant Blue 630. And so the first thing you can see is, yes, certainly we're not as bright, although hold that thought on what does that mean in terms of separating populations. But notably, we're incredibly quiet in the violet, right? And so what we've done is essentially, protect the violet, and in this case, protect the red, so you can actually leverage the fluorophores there. We're not contributing any spread there. Again, another thread we'll pick up is, yes, we want these clean fluorophores, but we also don't want to contribute spread anywhere else, right? And we've gone to characterize that using an Aurora as well. And then the third set of these slides is taking our Nova Yellow 610 dye, where we compared them to other dyes that have the same primary detector, right? So we compared it to Alexis 694, PECF 594, which is also called PE Dazzle, and then this ECD dye from Beckman. And you can see, right, we're actually brighter than Alexis 694. We're nearly as bright as this PE Dazzle dye, and we are brighter than the ECD dye. But again, something that a number of people actually pointed out to us was notably, look how quiet this is in the violet, right? So the spread contribution of these labels, right, is much, much less. So you have a very, very clean fluorophore that's contributing very, very low spread. And to that point, though, I do get a lot of questions, and I think that there's a lot of different perceptions of, well, how bright is it? What is brightness, right? And particularly because we can tune it, right? We think about brightness a lot. We think about brightness in two contexts. One, how does it compare to fluorophores that are out there now? And then also, can you separate biological populations, right? And I think we need to take those hand in hand. And so, with a lot of great suggestions, we actually started looking at separation indices, for the reason being that, this takes into account the 84th percentile of the negative, right? Because if you have a bright dye, but you're bringing up the background, right, then that's of no use to anybody, right? But what you really want is a very, very high separation of that population, and you keep the background very well controlled, right? And so we can see that in this context, our labels had essentially the same separation index, particularly off the NovaYellow, as APC, right? So we're able to separate these biological populations when we take a look at this. I think the other part of that, though, is that you certainly want something that's bright. You want to have that good separation, and you also want to protect your ability to use the other detectors that are on your instrument, right? Something that we'll pick up as a main thread in just a moment, right? And so when you actually take a look at the spillover spreading error that's contributed by these labels, right, the NovaYellow 610 was lower than 30 other dyes we measured on beads, right? And on an apples to apples basis, the Nova Blue dye was contributing much less spread than the Brilliant Blue, right? So the goal that we're striving towards here, and again, it's something that they're still working on, is how do you create clean fluorescent labels that don't contribute spread, but that can still give you very good resolution of the biology. And so to that end, we've thought more about, well, how do you think about upgrading a panel? And we'll sort of step this through. I've shown you some data off Symphony, off conventional cytometer. This is some work that we're doing quite literally right now, and then I'll show you some work we're doing with some of our partners and customers as well. But because of this capability, what we can do is take a look at what we would actually do practically with a conventional panel. Let's assume we were going in and doing some phenotyping, in memory T cells. And you can see, we would, particularly because we focus in on the BYG here, we've got 4, RA, 7, 8, and 25. But boy, we would really love to be able to get PD1 and CD8 on this, CD28 on this, but we simply can't, right? We've obviated our ability to do that because of the use of PE Dazzle, and we can't use Percy PSI 5.5 and PE Psi 0.5 together. So even though in this case on the Attune that we're running on, we have 13 detectors, we can only use 11. And so that's just not the case. When you move to this NovaFluor environment, you're able to stack those channels, right? So we can actually use each of these independently, right? And continue to use PE Psi 7 off of the yellow 4 detector, right? And so that's how we think about upgrading the capabilities of current cytometers. And so the work that's in progress there, we've shown, and I'll show some data around some of the 40 color work that we've done in upgrading the 34 color panel on a 64 detector instrument. We've done the same thing, we're doing very similar panel design work off the Fax Symphony, where we're going from 24, replacing 2, adding 2 more, right? Same thing for this, another Fax Symphony panel, and then our work where we're going in and upping this plex, right? And I think the point is that you can certainly increase the number of questions you can ask. You can get closer, we're stepping closer, I think, incrementally to the number of detectors we have. But the whole point is that you can actually lower the barrier on experimental design, because you can hit these detectors cleanly, and you can make the data analysis a heck of a lot easier. The second part of this is the notion of consistency and stability, and I think it fits, if you will, storytelling rule number 3, which is that surprise can equal delight, right? And I think this to me is very surprising, both from a couple different characteristics. One was, this is one thing that we had a lot of pushback on, that said, that's great, clean fluorophores. How do they work in practical life, right? Do I have to add any special buffers? Is there any special handling that I need to do? Can I put them in a master mixes? How hard can I push this platform? And so the answer is quite hard. And so we've actually tested this out to 3 weeks, but the labels are essentially bench stable. So we've just left this. It's probably one of the most simple experiments that we've ever run. We just left the labels out on the bench underneath foil, and then pulled aliquots off, right? And you actually see no difference across the spectrum when you see this. So it's 2 different modes of operating, right? One is accidentally leave something out on the bench, which happens, right? Or you're thinking around a high throughput experiment where you've got to run over many, many hours, and we're not seeing any change there. And for what it's worth, that DNA, that cartoon that I showed you, right, which looks like when it's laid flat, it's this cruciform structure that's 20 nanometers across, it actually has curvature because of the natural curvature of DNA, right? And so that provides a lot of physical stability, which both stabilizes the dyes on the platform, but also solubilizes it. It provides essentially a very protected water cage to that. To that end, we push the platform much harder, right? And so what we did is actually we actually left cells in fixative. And so how did we do that? We conjugated CD3 antibody to NovaYellow 610, stained those cells, and literally left them in 2% paraformaldehyde for 2 weeks. Honestly, we did this before COVID. We didn't realize this was a relevant experiment for that. We had to fix and then come back to your samples, but we did. Came back and compared it to fresh. we actually saw no difference in that staining. So we're extending this now to look at, okay, how many more, can you do this for multiple antibodies? What does that look like in a master mix basis? But I think really interesting result, and it shows the stability of this platform and just how hard you can push it from a practical perspective. The other thing that I think was a really important question that we started having was, okay, well, if you're relying upon oligo manufacturing and that's so consistent, well, what does that look like? And so we've been very open in actually showing our lot-to-lot data, right? And so this is actually our lot-to-lot data on manufacturing our Nova Blue and Nova Yellow, and we look at it across the spectrum, and we look at it in flow cytometry, and we look at it on cells. So I'm showing you here that just on, across the spectrum, less than 2% variation off the Nova Yellow and less than 5% off the Nova Blue, right? So our lot-to-lot consistency is just bang on. The label that you get now will be the same label you get, a year from now. And that's one of the reasons we're looking at this and tracking this, right, over time. The other part of this that's really important to us is that every time we ship a label out from our facility, we QC it on cells. So we take it, we conjugate it to an antibody, and we QC it on cells. We don't do it on cell lines. We don't do anything else, right? And so, we had really one of our first customers, and we handed them a data sheet that had that label that they had in their hand, QC on cells. And that was the moment where I realized, hey, I think we're onto something here with really addressing a frustration that we share, which is like, I want to see what I bought on a cell the way I'm going to use it. So the final part of this is a new to world capability, which is in this idea of controllable brightness, which we've proven out. I'm going to show you some of our data where we've gone from essentially 1x to 2x. We've also got some data where we've taken that all the way up to 4x and doing some work around high brightness. The way we're able to do that is we can take these networks and actually replicate them, right? And I think it's probably worth maybe getting ahead on a question is that while we're leveraging FRET, these are not tandem dyes. They do not degrade like tandem dyes. They don't need to be compensated like tandem dyes, right? So they are just FRET. The difference also is, remember, we're controlling the geometry of that FRET interaction, right? As I mentioned. But when we replicate that, we can take it all the way up to four clusters, right? And this gives us an ability to do very, very high brightness and eventually multiplexing. So we're investigating this now, both internally and then with a number of different customers, which is, well, what's possible when you can go that bright, right? But to show you how this works, we were able to show that we could replicate the network, but then also we could tune this up. So we've taken this one cluster label that we have from Nova Blue, turned it up to a 2 cluster. This is the FMO. So we actually just asked about FMOs. And then we actually doped in just the label alone just to see what the background is. Again, there's no special buffer. This is PBS, right? And then we can compare the 1 and the 2 cluster. And I think there's an important point to make when we actually look at this on the histogram is we do in fact get twice as bright. It's stoichiometric, right? Because in this case, we actually have 1 phyton on 1 antibody. And actually all the data I'm showing you is 1 phyton on 1 antibody. But we're actually increasing the separation index. We've increased the separation without increasing the background. And I think that's an interesting point. And it leads us to the ability where we can start thinking around, hey, how would I actually tune the separation index to antigen density? Because if I had a range of brightness that I could tune, we can start thinking differently around panel design. So hopefully I've shown you these sort of 3 different capabilities. And then, one of the questions was, okay, well, how can we characterize these labels and see how they interact with a number of other different types of labels? And I think one of the things that we were really striving towards is because we also have the ability to make new labels very, very quickly, again, something we'll discuss, because this platform gives us that lift. How can we think around also other labels that we'd like to go after, places in the spectrum we'd like to go after to enable some better biology? And the way that we like to think about instruments is they've got, so for instance, something like a Symphony, it's got 50 detectors. That's a lot of opportunity for colors, right? The stuff that's been published is about 28 colors. Aurora, same thing, 64 detectors. That sounds good to us. That's a 64, we should be pushing out towards that 64. The other part of this is, and I always like going back to this is, someone asked me in a webinar 2 weeks ago, well, why would you want to go up, 40 colors and beyond? And beyond the statistics, beyond correlation, I think one of the answers is, well, because it's there, right? And so what we did is we took some great work from Cytec which they've shared this 35 color panel and used that as a backbone. We took the 34 colors from that are commercially available, and we asked the question, which is, can we just drop in 6 of our fluorophores and do this on both high antigen density markers, CD19, kind of a gimme, right? But also low antigen density markers like chemokine receptors. And can we also use this as a way of looking at the impact that we have on spread across all these different labels, because that informs panel design across any flow cytometer, right? Across, you know, on a conventional flow cytometer, you're not going to be looking to use Brilliant Blue 15 and FITSI and a Nova Blue, right? But it certainly enables us to say, hey, if you're looking to use that Nova Blue, what's the contribution of spread across all of this? So it's a great instrument for looking at that for dye development purposes. And the reason we're able to do this and just literally plug and play these is because of the cleanliness of the fluorophores, right? It's a very busy plot, right? But we're able to hit those detectors very, very cleanly. And then this is also what enables us to go in and say, okay, well, if that's true, what does that mean? And the way we did that is we took the spillover spreading matrix with the backbone 34 color panel, right? And we said, okay, that's the foundation on where we're sitting on. And then what we did, Seddon on our team ran a really elegant experiment where she added one of our labels at a time and then saw, well, what's the impact on spread, right? And the goal here is what you're looking for really is the absence. You don't want to see a label drop in and then add spread across really anywhere in the panel. And that's what enables us to really look at this. And it has a big impact, not just on Aurora experiments, it has an impact on a Fortessa or Symphony or any of these other instruments. And so again, we dropped in one of these at a time and then calculated the delta SSM to say, okay, well, what's the impact, right? If this is I think Monica called it the perpetrator, what's the effect on all of the victims? The spillover spread, so what we did is quantify that, right? So what we found is honestly something that's rather predictable, which is that when we have a dye that's very, very similar, there's a very big impact on the spread. So as we would predict, spillover spread is specifically increased for BB515 and FITSI, but no other dyes are affected, right? It's what you're not seeing here that's important, right? So we're adding a blue dye and we're not adding spread anywhere else. Obviously very impactful for really all flow cytometry experiments. Little to no change off of the 610 or the 660, right? I'd contrast this 660 with PE Sci5, which is detected in the same primary detector, right? We're adding in a 660 dye and you're getting really very little contribution to spread. Nova Yellow 570, really all the yellow dyes behave honestly exactly how you'd expect, in the sense that when they're very similar to a dye, they contribute spread, but then nowhere else do they contribute spread, notably really anywhere in the violet, right? And so there's small changes in the spillover for PE when we just add Nova Yellow 570, a small change to the spillover for PE Dazzle when we add 610, and a small change in the spillover for PE Sci5. So it's very highly similar fluorophore, and you get a small change in that spread, but on a conventional cytometer, right, I think that it's even more impactful to think about you replacing these traditional fluorophores, because then you have this very low to no spread contribution into these other channels. So, another question that comes up, because we talk a little bit around, well, what is high resolution biology? And the answer is, the way we like to think about this is that when you can do that, hit these cleanly, have low spread, you get easier experimental design, higher resolution of biological populations, and then ultimately, something that you guys know I'm very passionate about also, which is easier data analysis, right? Because all the underlying algorithms, all the underlying manual analysis that we do all relies on this infrastructural problem we have with conventional dyes. To that end, we also, share this and try to make it understandable. This is a subway plot of the different phenotypes, which has had a life of its own as a visualization tool. And honestly, I'm trying to twist the arms to get someone to put this into an analysis platform. But it enables us just to look and peer into the data that we were producing, right? And, the important part for us was the phenotyping is great, right? In terms of just the ability to look at multiple phenotypes, that's fine. But what's more important here is that you're able to look at the deeper biology leveraging these NovaFluors. You're able to look at markers like CD40 and these chemokine receptors to really distinguish biological populations, right? And peer deeper into the biology. To that end, we've shared that white paper up on our website, but we've also shared all the data and all the analysis, right? Both the unmixed files and the raw data up on our GitHub site, as well as full repository. So the whole bevy of the analysis is all shared. How are we deploying that, right? There were some great questions that came up in talks around, okay, well, yes, but now what? And so this is the now what. The now what is that we have both human and CD4 label characterization kits, right? This enables you to characterize these labels on any kind of instrument. They're essentially the same, they're priced on a per test basis the same way as a BD or a BioLegend kit are. And again, we've got them across SK3 and RM45. The other part of this is our conjugation kits, and we've spent a lot of time vetting all different types of conjugation kits and really focused in on one that was incredibly simple to use, right? So the goal really for us was to be able to get a conjugation kit into a laboratory so that you can be running flow the next afternoon using our fluorophores. And then we pushed that a little bit harder, where it's only 5 pipetting steps and there's no special equipment. All you need is a centrifuge and a heat block, right? Which presumably we all, everyone has, right? And so, and the other part of this, the final part of this, was actually taking these conjugation kits, the 1x100 microgram kits particularly, breaking them out and making them competitive with antibody pricing. So a 1x100 microgram kit across any color is actually just 460 US, something we're also very open about. Another part of this is that, we've come out with a blocking solution that inhibits the stickiness that you see with monocytes and macrophages, not only for our labels, but also for all the Cy-based dyes, right? We're working to compare this now with other monocyte blocking solutions. We've seen very equivalent performance in experiments to date with that. And again, very open about enabling people with that blocking solution. Final part is, we do take on quite a few custom conjugation projects where, particularly because of that notion of the consistency and the stability, where we're able to do a meg and up of custom conjugation. And so, hopefully I've given you a sense of the future of what conventional dyes look like when we're able to do with NovaFluors, although you'll note, this line is twice as long as conventional, and we're not there yet. We only have 6 labels. But we've put a marker out there, right? For us, coming soon means at the end of June, we'll be releasing a whole new set of labels, and it'll be a larger number than the 6 we have now. So I'm really excited to share those, both what we're able to do off our current laser lines and then future laser lines. And again, we've always thought about in the context of how do you engineer and design fluorescent labels that have these same characteristics. So with that, if you've got any questions, please email me or email info@phytonix.com, and hopefully I have a couple minutes for questions.
Hi, Mike, that was absolutely wonderful. As usual, thank you. And we do have about 4 minutes for questions. So the first one is coming in from Joanne Lanigan, and she's actually, referencing back to Monica's presentation, where, so she said Monica presented on the complexity index tool from Cytec for assessing panel design. Did you compare the complexity index of the 34 color panel with your 40 color with your dyes?
We did, and I don't remember off the top of my head, and that's not okay. I should remember it. So one is I need to do that. I think it's a super good question with respect to the complexity that we know is being added from trying to do those 3 blue dyes together, right? And so I think, probably the better experiment would have been to drop out BB515 or FTSE on that and just use Nova Blue and one of those blue dyes. So, but I do not remember the complexity index off the top of my head, but we did run it. And we also looked at the similarity index of the different types of panels that we ran.
Okay. I have one personal question. So when taking into account spread, so something that people often suggest is to reduce spread due to the photon counting error, you can titrate down your antibodies. Since you have these tunable dyes, would you suggest titrating down the antibodies or actually tuning down the fluorochrome?
Yeah, I mean, you could really do either, right? Depending on what the application is, right? Because you could also choose, for instance, less bright, like you looked at the blue dye that's not as bright. You could say, well, I'm going to put that on a high energy density marker for multiplexing. Yeah, and we haven't moved to say, okay, there's multiple brightness levels yet that we've actually put out in product form. But I think it's an interesting point where you could say, for these very high energy density markers or something, I really want to say titrate down. I could use, for instance, a 1 cluster that we know is going to be stoichiometrically less bright. It's a really important point, and it's not something we've moved to fully explore yet, because I think we're still trying to understand if you could do that and do it at, if you will, 1x, 2x, and 4x brightness, and then you could also, for instance, change up the degree of labeling to have another axis of control on brightness. What would that mean, right? And what would it fit in terms of different antibody specificities you could leverage that on? I think it's early days for us in exploring that, honestly.
Okay, great, thank you. And then Su Hong asked, is the Nova blocking solution similar to FC block?
It is not similar to FC block. Good question. It's non-antibody based, non-protein based. It is more similar to something like the true stain monocyte block.
Okay, and then we have a question coming in from Rohit Mathur. He's saying that your NovaFluor dye analysis is done with a Cytec instrument. Will these behave equally as good with the BD Fortessa, particularly about spread? You have about 1 minute for that question.
Sure. Yeah. So I showed some data off there where I showed some off the Symphony. I think the Symphony is a really good analog to the Fortessa, and so that the spread bar graph I showed was off of that conventional cytometer. And so that should be a perfect analog for what the Fortessa looks like, because you just have more essentially have more PMTs, right? More detectors off of that symphony, but it's not, there's no unmixing occurring there, right? So the spread contribution, is going to be, it's still going to be a high performance dye on our Fortessa. That's something that we want to prove out from a multiplexing capability. So just because I'm opining on that doesn't mean that I've got data on a Fortessa that shows you can do that type of plug and play multiplexing, right? And I think it's very important. So that's something we are absolutely working on now, which is, okay, given that, what can you do? The limitation off the Fortessa is the trigon off the green, or off the blue rather, right? So there's not a lot of room to grow unless you're going to say, oh, go put an octagon off the blue. That's a stretch, right?
Yes. Perfect. Awesome. Thank you so much, Mike. We really, really appreciate it again. And that was a wonderful talk. Thank you.
Thanks.